start, and there's a few things I want to say before I begin the lecture. One is lab coats. So please, the only way you're going to get into the lab on Thursday is if you've got a lab coat on. If you're not, bad luck. Okay? We've got to enforce that law. It's already been 2 weeks where people have turned out without lab coats. So your lab coat must be on to go to the lab. The second thing to do with the lab itself is that the first 10 minutes, which means you've got to get in there on time, is the spot test, which will be reinforcing information you've learnt in the first 2 weeks of the laboratories. And the third one is an announcement that we sent out yesterday or the day before, that we have decided to do a light trapping evening. The weather is good this week, so Thursday this week at 7 o'clock. And it will be at the Horticulture Centre, which here is the building you're in at the moment. And then if you go up College Road a bit and then come in and park around here, around the CSRO building, walk up that road, we'll be in here somewhere. There's the demonstrators, Ray Lee will be running it, with mercury vapour light traps from 7 o'clock onwards, which will help with your collection. And as I said, bring some tubes, warm clothes, head torch if you need, net if you want, those sort of things. So that's 7 o'clock this week for light trapping. Okay, now let's make a start on lectures, and 2 lectures, one more on structure and function, and the second one serves as a pre-lab for your laboratory this week on the hemiptera. So that'll be a pattern that we'll now move into for the remainder of the entomology lectures. So really, by the end of this today, we will have done half the entomology content of this course. So 3 weeks already done. So we're going to get 12 lectures in entomology. Let's make a start with the idea around feeding and digestion. Okay, and this one might be a bit shorter and the other one might be a bit longer. I'll see how I go with time. Okay, we'll go back to Pete the grasshopper as our first visit, and we've already had a look at Pete once when we were looking at circulation, and we looked at the dorsal heart and aorta along here, and we also looked in that same lecture with these little white things, which is the respiratory system on the trachea. Today we're going to have a look at this multicoloured one, the yellow, the blue, the orange, the purple, the light blue. So we're going to look at the digestive system basically of insects. And we're going to try and learn from understanding that digestive system what we could predict about that insect if we were to find it, in terms of control and all sorts of other options. Okay, so what are the nutritional requirements of an insect? Well, they're not that different from most higher animals, but there are a few little points that I will bring out when we go through this one in particular. So the first nutritional requirement is protein. Protein's made up of amino acids. One of the problems that there is for some insects on certain plants is that they often lack one or more amino acids from that diet itself. So there are situations where they're lacking in those amino acids, and these are usually supplemented with endosymbionts that sometimes produce those, and we'll bring those into the lecture as we go through. But they're principally for tissue development, the development of new tissue. We use the protein to develop new tissue. Okay, let's have a look at the value. Now we've already got plants there. Let's have a look at plants as a nutritional resource by looking at the percentage of nitrogen, which is the building block of amino acids, so therefore it's a default measure for protein. So here we can have a look at a number of different diets that an insect may well feed on. The first one is xylem sap, down in the roots, typically, of plants. And you can see it's very, very poor. This is a logarithmic scale here, very, very poor in nitrogen content and therefore protein. And this isn't a surprise when we think about some of the things like cicadas. We say some of them have a 17-year life cycle, okay? They take a long time to get through on such a diet of xylem sap. Move up, we get a little bit better in terms of nitrogen content, but certainly a lot better in terms of sugar content with phloem sap. And then we have the pines, the gymnosperms. You can see they're getting up there a bit better than the flowering plants with leaves here. But if you really want to get to maximise your protein, you become a seed eater, okay? Which is logical if you think about it with vegetarians as well, but seeds are a key part of a human vegetarian diet. I guess in terms of leaves, there's also quite a preference for herbivores to feed on the new growing tips, which provide more nitrogen resources than the full leaf itself. But if you really want to get your protein, you become a carnivore. And as you can see up here, it's a scale above that of any of the other options. So I think we may have looked at this in one context already in the lecture, but we're now looking at it in terms of herbivory. We want to have a look at groups of or orders, and we already have started looking at orders. We looked at the hemimetabolous and the ametabolous this time round. That are pretty much herbivorous. So here's the first one here down the bottom. This is the stick insects, 100% are herbivorous. And there's always going to be little exceptions to the rule, and we'll bring some of those up at the time, but you know, as good as 100%. The Orthoptera, the bush crickets, the field crickets, the locusts, they're also herbivorous, a largely herbivorous group. And the other one that's very largely herbivorous is the Lepidoptera, the moths and the butterflies. So they're the three big herbivorous groups. And then if we want to shift down a bit, we have some of the bugs, which is going to be the content of your second lecture. About 90% of those are herbivorous. If you go at the other end, this is considered the most highly evolved of the groups, and one of the last ones we'll deal with in the labs and the lectures is the Hymenoptera, the ants, bees, wasps, the social insects. And you can see there, very few of those are herbivorous, only 11%, so very much gone for the carnivorous diet. Okay, so we move away from proteins and amino acids and take a look at carbohydrates. So what does carbohydrates provide for insects? Generally it's energy value, so it's your sugars, your nectar and things like that, and it's one of the principles of biological control for your natural enemies, particularly your parasitic wasps, is that you want to provide them with sufficient carbohydrate resources, that is flowering plants or supplementary sugar, so that they can actually live longer. So the more resources you get, the longer you live and the more energy you have for flying and moving around. So all good for them for that point of view. Generally stored as glycogen in the fat body, and the nutritive value of any particular carbohydrate does somewhat depend on the enzymes that an insect possesses. This is where we start to get a little different. Lipids. And with insects, they can't manufacture their own cholesterol. So they don't have a cholesterol problem like humans do, and get their blood cholesterol taken all the time to see if they're going to die from heart attacks. They actually have to get their cholesterol entirely from the diet, and they therefore store that as an alternative energy resource to carbohydrates. Some of the lipids are very important in their diet, though. One of those is for particular precursors for hormones, particularly ones that are involved with molting. So there are real value of lipids aside from an alternative energy store for insects. Again, we may have endosymbionts involved with some of these. The other little point I've noted down there on the bottom of the slide is that the needs differ according to the life stage and the sex of the adult. So the life stage, so when you're, say, a larvae, you require large amounts of proteins, fats and carbohydrates, because you're basically trying to grow as big and as fat as you can, as quickly as you can. So that's what you need in a larvae. But then if you're on a very high protein diet, and you've got a lot of those nutrients, you may not even need to feed as an adult. Okay? So there are many examples, particularly in the moths and butterflies and some of the other groups, where they've just got so much of their food resources already as a larvae that they don't need to feed as an adult. It's all stored with excess in their fat body. I also said there that there is some differences between the sexes, and the general pattern of differences is that female adult insects generally require protein, and they require protein to develop their eggs. Okay, that's if they don't come out with a full complement of eggs. Males aren't that interested in protein. They're obviously interested in mating, but they're interested in terms of food resources more for carbohydrates to get them around, move around, and to provide some longevity for them. So they're looking for different things. So your prime example of that would be mosquitoes, where the female mosquito sucks blood. Okay, she's getting that to develop her eggs. The male mosquito just flips around from flower to flower and takes a bit of nectar, so he's not involved with it. So we can get differences between the sexes in diet, particularly around production of eggs. The other thing there is that when we talked about hemimetabolous and holometabolous developers sometimes have exactly the same diet when they're larval and when they're adult. Classic examples of that would be things like lice and bed bugs that are basically feeding on the same thing. But remember, if we were holometabolous and you can have a pupal stage, we can completely redesign our mouth parts and feed on something different. So our nutrition requirements might change. Okay, let's have a look at feeding groups, and I want to do this using a table or a figure that's come out of sort of the recommended reading, which is Peter Cranston and Penny Cullen's book, Insects and Outline of Entomology. And they have a great diagram there, which looks at diet by a graphic representation. So here is a representation. We can either have a diet that is solid or a diet that is liquid. So a solid diet will include, eating plants, eating animals. A liquid diet would be sucking phloem or xylem from a plant, or indeed sucking things like blood from a host. And then we have in the middle of this sort of conundrum, and here it's on this axis here we look at a plant feeder and an animal feeder. In the middle of here we have a true generalist, and those true generalists are particularly things like cockroaches and earwigs. And we've got a few in between this sort of thing here where we get pollen, which is a sort of an in-between resource there with nectar. So bees have those mouth parts that they can take a bit of solid and a bit of liquid, remember, with the mouth parts that they achieve. Okay, so if we're to turn a look at the gut, how they're prepared for feeding on these diets, let's have a look at solid and liquid, I think, first. So this is what a solid gut structure would look like if you're feeding on something solid. It would be wide, straight, short, not a long gut at all. Very strong musculature, and key has abrasion protection. On plants, it's for rapid throughput of food, fairly rapid throughput. So it would be a fairly simple gut, and we'll go to gut structures in a moment, but there's a couple of things there we need to protect because you're breaking up either leaves or you're breaking up other insects and invertebrates you're feeding on, so you need to protect the gut with abrasion protection in there. And the other is it's fairly simple, okay? Wide, straight and short. Okay, if we're a liquid feeder, you have a very different problem. You might be feeding on xylem or phloem, but you have less nutrients in those, so you've got to extract them. You've got a lot to process through in order to get equivalent amount of nutrient. So therefore you have a very long, narrow and convoluted gut, which then enables more time for it to process through the gut, which means you can extract more nutrients with time as it takes longer to travel through there. So it's maximising the time in contact, and it has no abrasion protection because a liquid diet is not going to have an issue with it. So if you were to open up insects straight away, if you didn't know anything about that pest that you found, you'd have a look and you'd say, yep, I can have an idea of the diet, as you can with what we've already said about mouth parts. Okay, let's look at a plant diet, and these all interact when you put these together. A plant diet, they're just sitting on something. They're eating, and they're on that all the time. If you're a caterpillar, you're basically on that plant for most of your life, unless it runs out of resources and you might move on to another one. But it's short, you don't have storage areas because you're just continually feeding, and you're really trying to get through large and large amounts of food because, remember, there isn't a lot of protein necessarily in plant diets. Last option here. If you're on an animal diet, it's very different because your intervals between meals may be very irregular. Okay, so on an animal diet, you're going to need storage capacity. So either something to store if you're a predator or something to store blood if you're actually a blood feeder. Okay, so these are the sort of interactions you can see under how the gut could be assembled. The other thing I want to look at when it comes to feeding in insects is to look at how they feed, the behaviour during feeding, and I'll use a number of examples to illustrate those as we move through this table and revisit it. Oh, no, I don't need to revisit it, I've got only one picture there to show you. So we'll start, let's say, with leaf feeders. Here, I know it's a small table to see, but this is a locust here, and this one here is a tobacco hornworm, feeds on tobacco. So here we can see its diet of both leaves here. They're fairly big in weight, you know, they're fairly large things sitting on those plants. Their meal size is somewhere between 2 and 17% of their body weight. And their meal duration, though, is very short, only 6 to 8 minutes for a locust, and then it spends 70 to 80 minutes not eating. And for a tobacco hornworm, 10 to 25 minutes and 15 to 25 minutes eating. So if we look at the percentage of time feeding, less than 10% for a locust, and somewhere sort of in that 50% range for a tobacco hornworm. Okay, so why, if you're sitting on your food resource, if you're a caterpillar, do you not spend all your time just trying to maximise the food that you eat, the amount of quantity that you're getting? Or if you're a locust, they will probably move between more than one type of plant, as they're much more mobile than a caterpillar is. So why are they only spending these very short time on meal durations? Well, the general theory, and we won't do much of that within this course, is that plants fight back. They have defensive toxins, and those are either direct toxins or things that inhibit the digestibility of what they're eating. So if you take a diet for a period of time and you feed on that, then you require a period of time to get rid of those toxins and those overcome the defences that digestibility reduces. So you're not spending all your time feeding. The other thing that's relevant to feeding when you're out exposed, like say a caterpillar is that you are exposed to natural enemies. Okay? Because when the actual active feeding produces volatiles, and these volatiles interact with the saliva of who is feeding, or the species feeding, to produce a message out there for all your parasitic wasps, for your predators, come along and feed and eat me. And believe me, there is a high probability that you will be parasitised or eaten. So the act of feeding is fairly risky. So you don't want to spend all your time feeding, okay? So you've got to actually, for two reasons, to digest it and to not continually be at risk of being killed. Okay. Let's have a look at another diet here. This one here is plant fluids. Hemiptera, this is a little brown rice leaf hopper, and it's have a very small weight, 2.5 milligrams, but it feeds continuously, okay? 85% or more of its time is feeding. So these things have basically got their mouth parts plumbed into one point, particularly with scale insects, this is the case, and leaf hoppers, they can move, but then they spend most of their time just sitting there and feeding. And this creates issues when I talk about exposure to natural enemies, and we'll talk about that a bit more in the Hemiptera lecture. So it does have risks doing that sort of thing. These are a couple of diets here that are nectar. So we have a butterfly and sheep blowfly. You can see here they vary enormously in weight, but they take not large percentage of their body weight in a meal, and their meal durations, again, have a reasonable time out. That's just because nectar is a ephemeral resource. They take a bit energy, go back and get more. It's not really something that they're going to be able to spend a long time on a flower, because the flower is not going to reward them with enough nectar to completely achieve any needs for the day. Last one here that we want to look at is, we haven't got a predator in here, but we have got Rodnius, which is, hang on, where is it? Oh, here's a predator. This is an assassin bug here. We can see here with this assassin bug, weight's about 105 milligrams, about 20% of its body weight. Spends a lot of time feeding. It doesn't go off and, it's not going to be attacked by other predators. And a lot of time between meals, greater than 24 hours. And then if we have a Rodnius, which is also a species of bug, which we'll visit during these lectures, that actually sucks blood, it spends 40 milligrams, but its meal size is 300 milligrams, so it's 750% of its body weight it takes in one sitting, okay? And days in between. So this is a Rodnius after it's had a feed. So here's one here, and here is after its blood feed. It's basically increased its body weight by 750%. So it's a glutton, if you like, but then it can spend a long time out. This is the same principle that we've used for mosquito control, because mosquitoes are very similar. They take a large percentage, the female of blood, of their body weight, and then they move off. So when you look at control of mosquitoes, They often go to wolves, rest for long periods on wolves after they've had a blood meal. Doesn't help you, you've been already attacked by a mosquito, but it will mean that they're more exposed to insecticides on such areas when they're coming back. The other thing that's relevant to feeding and digestion and food consumption is behaviour, is just to look at how this might vary within an instar and between instars. So this top graph here is the time between an instar, so a molt here and a molt here. This is day and night feeding, and in this particular example, more feeding is done during the day, but they're also feeding at night. Caterpillars would be a case in point. But what you can see is it's not consistent from molt to molt. The peak in feeding is actually in the midpoint, if you like, between molts. So if you were trying to do bioassays and you wanted to look at toxicology of certain pesticides, you wouldn't want a newly molted insect or a about to molt insect. You want insects around this point here that are going to take in more of the food. Similarly, this is 3 different insect species, but they all show the same pattern: cabbage white butterfly, African plague locust, and Rodnius, the assassin bug that takes blood. And you can see all of these are 5 instars. The last instar is the stage where most of the food is consumed. So over 60%, and in general the rule of thumb would be over 50% of all food consumed during the lifetime will be consumed in that last instar. So if you want to control insects, you've got to get to it before the last instar, okay? These instars much better, because by the time you've got it to here, they're already taking out a large amount of the crop material that you may wish to protect. Okay, I mentioned already, I think on a couple of occasions, that we're understanding more and more about bacteria and other symbionts that may exist in insects. And in the case of diet, symbionts certainly play certain parts in providing anything from sterols, vitamins, carbohydrates, amino acids, okay? So a large range of symbionts do exist in insects. I think probably at least 70% of all insect species seem to have at least a symbiont associated with them. Okay, so it's majority by a long way. The type of symbiont that we might have living inside an insect can be bacteria, bacteriolite, yeast, fungi, protists, you name it, they all exist. Okay. What we want to now look at though is where they might exist in the insect in particular. So the location. They can be free in the gut or they can be in certain locations. So let's have a look at some of these options where symbionts might reside. Okay, we've got a bunch of different feeding diets here again. Here's wood, here's a green plant, here's phloem, vertebrate blood, and that's a detritus feeder. So if we look at wood, we've got here cockroaches, termites, and some boring beetle species, as well as sawflies. And all of these have microorganisms associated with them, typically in the hindgut is the location for these. Could vary, flagellates, fungi, bacteria, yeast, they vary in what it is. But mostly, for wood feeders, they're around cellulose digestion, helping break down cellulose, which is not an easy product to break down without something like that. You can see most of those around protein, nitrogen fixation, etc. Green plant here, we have a particular insect. Well, obviously wood, I've got 2 examples where they might be external. Is wood products, these 2, or with wood in particular. So here have a look at wood. This is bark beetles, and they carry fungus in a specialised tissue, which is situated just here. Between the thorax and the abdomen, and if you pull that apart, it's this little tissue that sits there exposed between the thorax and the abdomen, and it's where the fungus is carried, and they bore into trees, and then that fungus comes into the trees, and that fungus helps with the breakdown of the wood products so that its larvae can actually break down the wood. So these are externally on the beetle, if you like, on those little mycangia on the beetle. Another one here with green plants as a diet and microorganisms is these particular ones, actinium, which are leaf cutter ants, okay? And again, the position is external, ecto, fungus, cellulose digestion, and here's an example here of leafcutter ants. So there's about 43 species around the world, mostly concentrated South America, Central America. Here they are cutting up leaves, taking them back to their environment, and you can see here cultured on those leaves are fungi, and the fungi provide the food resources that they need, not the leaves themselves, the fungi they grow on them. So externally growing those symbionts that they need for diet. Have I got that one back again? Go backwards on that one to this title here. Again, we've got phloem, which is said is very poor in nutrient resources for protein. So we've got bacteria producing amino acids, or in some cases sterols, which they can't produce. And in vertebrate blood, it seems that most of them associated with vertebrate blood are associated with getting them B group vitamins. Okay, so that's the sort of general picture for endosymbionts, or ectosymbionts as they are within insects for digestion. Okay, the other thing that needs to be considered is how are you going to get your symbiont from you to your offspring, or from you to your siblings, or you to your other social insects in a colony if you're a colonial species. Most of the transmission seems to be transovarial, so it goes through the mother. So they're either external and she's laying the egg, or internal, or both, to the egg, and that gets it through to the offspring, and they continue from generation to generation that way, 70%. But if you've got social or subsocial insects, we're talking here bees being social, subsocial being things like earwigs, cockroaches, and a few others, they sometimes do things by slightly different. So they either eat excreta that might come from the mouth parts of someone in the colony, or from the anus of someone in the colony, or eat the old shed skins or molt skins of those in the colony, and by that way you get transmission through the colony. Okay, we now want to look at gut structure. And some of the terminology in gut structure is really simple, so we'll start with a simple start of that, and then we'll look at it also in relation to vertebrates and what we might see in ourselves when we look at it in comparison. That's good. Okay, so there are three main regions of the gut: the foregut, the midgut, the hindgut. They can't be too hard to remember. The foregut is ectodermal in origin, so it has cuticle associated with it, and it's primarily responsible for ingestion, getting the food in, and storage. The midgut is endodermal, so it doesn't have cuticle associated with it. It's responsible for digestion and absorption. And the hindgut, again, like the foregut, is ectodermal, so it can have cuticle associated with it. It's involved in water relations and the formation of waste material. So that's not surprising: ingestion, storage, digestion, absorption, formation of waste material. So if we're to look at the human equivalent of the foregut here, we've got the mouth, the esophagus, and the stomach for storage. Okay, so that's our foregut. And then when we come down to digestion and absorption, it's the small intestine, where all digestion and absorption takes place, and then we get water relations and excretion of waste products is the large intestine. So we've got functional equivalents, if you like, in ourselves to what insects have. Okay, let's have a look at that when you put through a cross-section of an insect. And here's a cross-section of a generalised insect. We want to start with the foregut. So we can see here we've got the preoral cavity where all the food comes in, and we've got a pharynx. Now the pharynx is a musculature part of the head, and if you're a fluid feeder, you often have a quite musculature pharynx because it's got to actually suck up, get enough pressure to suck up fluids. The esophagus, just a tube as we are with ourselves. The crop is here, and crop sizes vary according to diets, and we'll look at that over the course of this lecture. Then the area called the proventricular. So we'll look at this area, and this area. So the crop, one of the advantages of being the origin that I said it was, which is, if you remember here, ectodermal in origin, is that the crop can be lined with cuticle. So that means that if there are toxins in the diet or things that you don't want to leak out while you've got it in storage, it can help with the fact that you've got like a plastic bag there, if you like. Nothing's getting out, nothing's getting in. So a good means of storing it. As I said, it does vary in use and size depending on diet, with obviously intermittent feeders needing larger crops. If you have a solid diet, particularly, well, no leaves and also, say, other invertebrates that you're breaking up, the proventriculus has what's known as cuticular teeth in it, so little grinding things. So as it goes through there into the midgut, it's grinding and grinding up the food, so it's getting to a smaller articulate side when it's getting through there. So that's the example of the proventriculus. There's a little valve here at the end which then lets it through. The other one that's interesting is the salivary gland, and we'll visit this on and off during various lectures, but the salivary gland obviously just helps with the breakdown of food, but it can do that in a number of different ways. It can either just provide some to the host while it's just normal chewing, or it can do it externally. So this example of external use of the salivary glands here come in two instances. There's a cartoon down there which says, I worked hard on this meal, the least you can do is regurgitate on it. So we're talking about flies that are putting down salivary secretions to help sponge up the food with that sponging mouth part. But the other example I want to use, or two examples I think I want to use here, this side of the next, is in the family, well, in the order Neuroptera, the lacewings. We have antlions here. And they have, they're predatory, you may have seen them. They lie at the bottom of little pits they dig and attack anything that falls into that pit. And they have their mandibles quite long, quite vicious here. And then they'll inject, once they actually get hold of a prey, they'll inject salivary fluid into it and then make basically the internal contents of what they've caught soup. And then they'll suck it back out. So it's an external use of the salivary gland to break down the products of the host. This is another one that we'll revisit these two next slides in the Hemiptera lecture. But some of the Hemiptera also have a method of feeding where they actually regurgitate down onto the plant and then suck up the contents. Again, external saliva. And some of the Hemiptera, and we'll revisit, I said, next lecture, so I won't spend a lot of time on this part. Also, this is like an aphid, actually can plumb their mouth part in between the cell walls all the way down here to get into the phloem. So they're not puncturing and rupturing anything, but what they actually do is sheathe that stylet as it moves through with products of the salivary gland, which then help it not be so exposed to any defences the plant might mount against its mouth part being in there. So it's like putting a sausage skin over it. And when you pull it back out, you can see exactly where it's been feeding, if you've got the right cross-section through the plant. This is just another version of that. And I want to really move on to storage and crop levels, but I now realise I do have a picture here of the proventriculus here, which is just this little bit here, and you can see it has internal teeth, which I mentioned for mashing up food products. So that's the proventriculus in solid feeders. Storage, though, in the gut can be in various areas. So we're talking about crops, and we're looking for where it's going to be. So here in a short, straight, wide gut caterpillar, we can see the crops just along there in the foregut, nothing special, nothing large. In a butterfly, it's also in the foregut, but it's made more into a offshoot, if you like, of the foregut, where it can actually store it. And here in the fly, we have it again here, and these are both sort of nectar storage, if you like. If we get to some of these other groups here, it does tend to vary to where storage in the gut is. So Micrasia is a sawfly that feeds on oils, and well, it has oils from resins and things from pines that it will feed on. So it has a little crop here and a diverticulum which stores all the regurgitated oil which it uses in defence. Then we've got beetle larvae, scarabs. So remember we saw that body form last week, scarabeiform larvae. They have fermentation chambers full of symbionts, so do termites, and these are down in the hindgut region, similar to what a cow has, and we'll talk about that when we move through. And this is the blood feeder, and it's using the midgut for storage, and that makes some sense because it wants to actually extract things out of that midgut when it's sitting in there. Okay, so I think that one we have already sort of gone through in terms of B vitamins and things like that. So let's move through. Oh, well, I can actually use this slide by slide, is what I've done. So if we go back through and look at, let's see, okay, Coleoptera, the beetle larvae and the lower termites. So these wood feeders, and what I'm interested now is where the endosymbionts might reside, not just where the things might be stored, but where the endosymbionts that help break down things. In this case, this is cellulose digestion, and here we have Coleoptera scarab beetle larvae and termites, and you can see in termites it's in the hindgut, they're all residing through here in these speckled zones, and in scarab beetle larvae the symbionts actually are both in the midgut and the hindgut, so two regions there. So it's varying, they're in specific locations, and these are like in the case of termites, and to an extent with these, they said like the rumen of cows, where they're just basically sitting in there and fermenting with those endosymbionts to try and break down cellulose. The next one I want to bring in is phloem feeders. So here we have these phloem feeders, they have bacteria, they want amino acids. So where do the phloem feeders store their endosymbionts? We've got here in the hemosyl. So here we can see an aphid, and we can't see it anywhere inside the gut itself. It's sitting outside the gut in the blood. Okay, so a slightly different region. Phloem feeder external. Next one here is blood feeders. So we have the blood feeders that want B vitamins. Let's have a look at where some of the blood feeders keep their endosymbionts. Well, in the case of the assassin bugs that are blood feeding, in this case it's known as a kissing bug. I'll elaborate that in the next lecture. We can see the endosymbionts are sitting in the storage area, which was the midgut. And it said that makes sense, that's where you've got to actually have that blood products to get the amino acids and things that you want, or the B group vitamins out of there. This one is just to say that it varies then with the last options here where symbionts might be. We've got them in caterpillars in both the midgut and the hindgut, in flies in the midgut, in locusts and grasshoppers, as we saw in earlier ones, right through the whole area, but it's where the storage areas are. And in a honeybee, it's in the hindgut down here. Okay, so the position and locations varies, but it typically, I guess, relates to where you want to extract the products that you need and where it might be stored to extract those products from. Okay, let's take a look now away from endosymbionts and look at the midgut in particular. So the midgut is obviously between the hind and the foregut, and there's a couple of structures that we can see there. There's ceca right attached at the point between the foregut and the midgut, and then there are something at the other end called Malpighian tubules, which we'll also talk about. But let's have a look at the midgut first. The midgut actually has its own internal, like, sausage skin within it, and this is known as a peristrophic membrane. So as the food goes into the midgut, it goes into this sausage skin or peristrophic membrane, travels along here, comes out at this end, and then can go backwards through here. So the peristrophic membrane is believed to help on a number of fronts for certain diets in particular. So it has a protective function. So it means that when it first gets into that midgut, if there are any still abrasive things or toxins or pathogens, There's time sitting in that peristrophic membrane for it to be able to break down and not get out into the rest of the hemosyl and other products in the inside the insect. It also has a value in partitioning it. So if this was just one big long thing, it would just push its way through quickly. But when you've got this here, then it can come back the other way and move around, etc. So it enables to partition it, which gives further digestion time. Remember, that time sitting in the midgut is important because that's where your digestion and absorption is going to happen. So having it extending more time in there is useful. The other particular thing that you can see here with midguts is that they have the anterior region gastric ceca, and these can vary in number and quantity depending on the insect itself. And these just provide more surface area for exchange of nutrients from the food out to the hemosyl. So here we've got an example of one here. So we can get these bathing the hemosyl more and getting more surface area for exchange. The last thing that's interesting about the midgut, before I go to one variation on that, is that its pH is generally 6 to 8, normal, but when you look at leaf feeders, the pH is actually very alkaline, like 9 to 12, and the possibility that that pH is so high is believed that it's involved with breaking down, again, plant toxins in the diet. Okay, we have now the example of phloem feeding, and here's an aphid, got its way down into there. And just look at the gut of these phloem or filter feeding type insects. Now remember, phloem has lots and lots of sugars, but not necessarily lots and lots of protein or amino acid. So you've got to try and lots of water. So you've got to try and pass through lots and lots of water and other things to get what you need out of it and get rid of too much sugar as well. So how do you get rid of your carbs and your water? They have a system known as a filter chamber. And what they're doing is they actually wrap the beginning of the hindgut around the anterior of the midgut. And by doing so, that means as soon as the food products reach the anterior of the midgut, the hindgut is starting to extract water out of it straight away, rather than going through the midgut to the hindgut to get there. So it's a super extractor, if you like, of taking out water and carbohydrates out of that diet. The end result of that is that you'll see that these sort of feeding insects, and we'll bring that back in the micro lecture, produce a very sugary secretion known as honeydew. And we'll talk about that in the next lecture in terms of the consequences of honeydew. Okay, I think I'm moving into the hindgut now. So I said this might be about 5 minutes short, which is what I planned. And looking at the hindgut, and well, actually we've just got to that junction here now, and we've seen the Malpighian tubules. So let's have a look what the Malpighian tubules are. There, remember, there's lots and lots of these projections that are sort of snaking their way around the hemosyl of the insect. Okay, they vary in number from about 2 to 250. So locusts, which have a very varied plant diet, moving from one plant to another, have 250 Malpighian tubules, so a lot more. They provide high surface area because they are actually long threads going into the host. Seeing all my notes right here, I'm going way ahead of that. Let's see if there's anything to say. They are the equivalent of the kidney, if you think of it in our thing. So they're involved with the same functions as the kidney. So they're actively pumping potassium ions to get an osmotic gradient so that you get electrically positive inside. So water, waste products and amino acids passively flow in along with potassium, and it gets a primary flow of urine into the gut. Okay, so if we look at that on a schematic version of it here, we can see the Malpighian tubules here, and all these nitrogenous wastes, water, amino acids, sugars and salts are going into the Malpighian tubules. And then when we come through here into the remainder of the hindgut, which do I say, yeah, I'll leave that little bit and come back to that, the remainder. So they're coming in through there. This is a dissection of a cockroach gut. Looking at it from this end, this is the beginning of the midgut, and you can see all the gastric ceca here, and then you can see the midgut. So you would say this is a very varied omnivorous diet with all those gastric ceca in there. Then you can see a lot of Malpighian tubules floating through here, and this is the hindgut where you can see some of the solid waste being started to be packed as it moves through the hindgut. The other parts of the hindgut we've got there are the rectum, the colon, the rectum and the anus, and I just want to talk about the ileum, the colon, the rectum and the anus. The ileum's just somewhere where maybe symbionts, if we do have those symbionts, reside, as we saw in hindgut in some of the insects. The rectum, though, has rectal pads which help conserve water and sugars before they're dumped out. So, and some inorganic iron. So if we go back to here, with this slide here, remember these were taking out nitrogenous waste, water, amino acid, sugars and salts, but then the rectum is starting to absorb them back in again. So, you are not losing as much water. So the water comes through, the rectum is taking them out again. So it's taking out some of the sugars, some of the amino acids and the water. So this gets it one way, to get it all flowing, and then you get saved and conserved water through the rectal pads, which we say there. So they're working together, if you like, Malpighian tubules and the rectal pads in conserving the resources. And the main thing that you want to conserve is water. You don't want to be too dehydrated in the process. You want to get the maximum water out of there. So that when you've got a really low water diet, like you're a stored grain feeder, they actually wrap their Malpighian tubules around the rectum, and they have a thing called cryptonephritic systems, which extract even more water out of the diet, so that their actual pellet they produce is just dry. There is no liquid at all in the pellets that they're producing. Insects don't excrete urea like we might. They excrete uric acid like birds and reptiles do, so that's a bit more water conservative in that process. But it does produce ammonia, which they must flush out a bit more. Last slides here look at a couple of other excretory modes in one other area of the gut. It's worth mentioning. That is the fat body here. So this is a cockroach that's been dissected, and you can see all this white stuff. This fat body sitting in the abdomen of the cockroach. And the fat bodies are the equivalent of the liver in our system. In our system. So the fat body is intermediate metabolism, metabolism of carbohydrates, lipids, nitrogen, storage of glycogen, fat, protein, and synthesis of particular hemolymph protein. So it's acting essentially like the liver is in ourselves. So metabolism of all those particular products in the liver. The other last one I want to say is in some insects they don't actually excrete anything out of their anus for a long period of their lifetime, and this happens in a couple of particular unique instances. If you are a parasitoid larvae sitting inside a host, you don't want to dirty up the environment inside the caterpillar that you're living in. If you're a social insect like a bumblebee here, you don't want to dirty up the cell that you're living in by excreting in it. And also it's the case with these antlions who don't also excrete because they don't want their hidden area to be probably picked up by insects that would otherwise know that they're there through the chemistry. So the uric acid is inactivated and it's stored in cells in their fat body, and when they emerge as an adult they produce a large meconium pellet, which is basically the build-up of all their excretion during their immature stages. Alright, and that finishes digestion and excretion. So we've got, I think I can get through the other one. So we'll make it about midday. Might be a little bit in front of midday for the second lecture.